Hi everyone, my name is Nimeesha Komatireddy, and today I'll be presenting my research on how retinoic acid receptor gamma, also known as RARG, modulates ferroptosis in doxorubicin-induced cardiotoxicity. My mentor was Miss Nassim, and she was able to help me identify my project idea as well as with my team member who's actually here today as well, Marlena. Doxorubicin is a widely used chemotherapy drug, but its use is limited by its potential to cause heart damage. My work focuses on understanding the molecular mechanisms behind this effect, particularly the role of ferroptosis, which is a form of regulated cell death, and how genetic variation in RARG affects cardiomyocyte vulnerability. So before I get started, I just want to give a little introduction about myself. I'm a current student at Arnold L. Beckman High School in Irvine, California, and I'm a junior who's really passionate about cardiology, medicine, and data science. I serve as the president of OC Coder and president of Mindtricks California, which is a nonprofit that introduces youth to brain science, and I'm involved in several other STEM outreach initiatives. My long-term goal is essentially to become a physician scientist where I can integrate research with patient care to help improve health outcomes on a global scale. So, to start off, doxorubicin is effective in treating many types of cancer, including breast cancer and leukemia. However, a significant limitation is its cardiotoxicity, which can manifest as heart failure or arrhythmias in some patients. Ferroptosis is a type of cell death that occurs when iron accumulation drives lipid peroxidation, ultimately damaging cell membranes. And RARG is a nuclear receptor and a transcription factor that regulates gene expression in heart cells, and it may modulate how susceptible cardiomyocytes are to doxorubicin-induced ferroptosis. So this study had four main objectives. First, we wanted to explore how RARG influences ferroptosis in the context of doxorubicin exposure. Second, we compared gene expression responses in wild type cells, RARG corrected cells, and cells carrying the S427L mutation. Third, we aim to identify ferroptosis regulated pathways, and that could serve as therapeutic targets. Finally, this work lays the groundwork for personalized strategies to reduce cardiotoxicity risk in patients undergoing chemotherapy. So, ferroptosis is distinct from apoptosis or necrosis or essentially just any type of cell death pathway because its hallmarks include accumulation of reactive oxygen species, lipid peroxidation, iron overload, and mitochondrial dysfunction. Morphologically, ferroptotic cells show cell swelling and a dramatic reduction in mitochondrial volume and cristae. Molecularly, ferroptosis is regulated by antioxidant genes like SLC 7A11 and GPX4, as well as proferroptotic enzymes like lipoxygenases and NAPDH oxidases. Classic ferroptosis inducers, such as elastin and RSL3, can trigger this pathway, making it a relevant mechanism in drug-induced cardiotoxicity. So doxorubicin kills cancer cells by intercalacting DNA, inhibiting topoisomerase 2, and generating ROS. However, these mechanisms also harm healthy cardiomyocytes. RARG is a transcription factor that is crucial for heart development and cellular homeostasis. The S427L missense variant disrupts RARG's F domain, and this reduces its ability to activate DNA repair and antioxidant genes. As a result, cardiomyocytes carrying this mutation are more vulnerable to oxidative stress and ferroptosis during doxorubicin treatment. So essentially, to investigate how RARG modulates ferroptosis, I analyzed two publicly available RNA sequence datasets, and these were curated ferroptosis-related gene lists to explore transcriptional signatures of ferroptosis under different experimental conditions. Specifically, I analyzed the GSE 164637, which is a RNA sequence of a model of doxorubicin cardiotoxicity that provided clear or case control and treatment group relevant to chemotherapy-induced cardiac injury. And GSE 181517 was also another sequence which compared wild type and RP mutants treated with or without doxorubicin to evaluate ferroptosis linked transcriptional responses across genetic backgrounds. And both databases were downloaded from the NCBI Gene Expression Omnibus, which is a peer-reviewed open access repository that provides raw expression counts necessary for custom downstream analysis. Essentially, how this worked was we curated ferroptosis-related genes from FerroDBV2, which is a gene that's also associated with ferroptosis but without known regulatory effect, and standardized expression data using log-based 2 transformation. Differential expression and interaction analysis allowed us to identify how RARG genotype influences ferroptosis gene transcription during doxorubicin exposure. So in ROG-corrected cells, the doxorubicin treatment induced stronger antioxidant responses, which activated genes like the SLC7A11 and QO1 and CHAC1. And these genes enhanced glutathione synthesis and protect against oxidative stress. At the same time, genes involved in lipid peroxidation, these genes also genes that were or they were also protected against oxidative stress. And at the same time, the genes that were involved in lipid peroxidation and ROS production, such as the PTGS2, ALOX1B, 12B, and NOX5, were suppressed. This suggests that maintaining RARG integrity reduces ferroptosis susceptibility and protects cardiomyocytes. In contrast, cardiomyocytes with the RARG S427L mutation showed amplified ROS generation with increased expression of genes like NOX1, NOX2, and the AKR1C family. Interestingly, some lipid oxidation genes were less responsive, but overall, these cells exhibited higher oxidative stress and ferroptotic pressure. This provides a molecular explanation for why patients with this variant are more susceptible to doxorubicin-induced cardiotoxicity. So these results highlight the central role of RARG in modulating ferroptosis in wild-type or RARG-corrected cells. Antioxidant pathways dominate, protecting against ferroptosis. In S427L mutants, ROS production overwhelms defenses, and which leads to an increased cell death. This tug-of-war sort of relationship illustrates how genetic background shapes cellular responses to chemotherapy and explains variability in patient susceptibility to cardiotoxicity. So essentially, these findings suggest that targeting ferroptosis pathways could reduce cardiotoxicity without compromising chemotherapy effectiveness. Activating NFR2 or enhancing system XC transporters are potential strategies. Moreover, screening patients for RARG variants could also allow personalized therapy, optimizing doxorubicin dosing in order to minimize heart damage. While these results are promising, there were a lot of limitations. A lot of these bulk RNA sequences were not able to capture a lot of the cell type specific effects because it essentially it was also not able to show how the gene expression wasn't able to reflect the protein activity. Future work should also integrate single-cell transcriptomics, proteomics, and functional studies in vitro and in vivo to validate these findings and translate them into therapeutic strategies. So in conclusion, our study demonstrates that ferroptosis is a key driver of doxorubicin-induced cardiotoxicity and that RARG plays a protective role. Maintaining RARG functioning enhances antioxidant defenses, whereas the S427L variant amplifies oxidative stress and ferroptotic cell death. Importantly, we identified molecular targets such as SLC7A11 and NRF2, which may guide the development of cardioprotective therapies. And ultimately, these findings support the use of personalized approaches to minimize cardiotoxicity while preserving chemotherapy efficacy. Here are the references that I utilized. As for my acknowledgments, I want to thank my mentor, Ms. Nassim, who's also who did research similar to this in Simon Fraser University. And I also want to thank my partner, Marlena, who was also there with me during this project. And I also want to thank Talaria Summer Institute for giving me this opportunity to produce this research along with my partner and my mentor. Here is just my contact information, and thank you.